Hi guys, welcome back to the channel. Today I'll give a focused summary on an article recently published in the Science journal talking about a potential new target for fighting the coronavirus. If you are new to the channel, I'm Dr. Han. I'm so happy you're here. Now in this channel, you will find science review video, updates on the latest global health problem. I also like to share tips and tricks for students learning academic and personal development. If these are your interest topic, please consider subscribing to the channel. If you have already subscribed, thank you very much for coming back. So this past week, I've read a very interesting article published in the Science journal talking about finding a new potential target that can be used to design new antiviral therapy against the SARS-CoV-2 virus. Now in this video, I'm going to take a deeper look with all of you in a more layman term, okay? Without further talking, let's get started. So today's topic is called potential novel target for antiviral therapy. Now this is a very early stage of investigations. Hopefully we'll see more in the near future. Alright, first of all, a disclaimer. This video is my interpretation summary of publicly available scientific information. This video is not serving as any advice on treatment, diagnosis, and preventions of any diseases, okay? And there's no affiliations in this video. Now, let's have a little bit recap of how the SARS-CoV-2 virus binds to our cells. Okay, here on the left-hand side is the viral particle, which is full of or covered with the spike protein, okay, those mushroom head particle or figures. Okay, now in fact it has two parts, okay, the part that it's actually interacting with our cell. First is the RBD, okay, the receptor binding site on the mushroom head, okay. Now, this mushroom head actually belongs to the S1 subunit of this spike protein, and in fact, the stem, we call it the S2 subunit, okay. Now, this S1 subunit will bind to the ACE2 receptor on our cell. Now, here is a figure showing you how it interacts with the ACE2 receptor on human cell, okay. Now, but only interacting is not enough to gain cell entry. In order to gain entry, it needs to be first cut, okay, at a junction between the S1 and S2 with a protein called furin. Now this furin is expressed on a lot of our cells, so that's one of the reasons why SARS-CoV-2 can infect many cell types in addition to our respiratory epithelial cells, okay? So here are some of the new findings that is reported in this article here. Now, in order for it to gain entry, they are looking at a new cofactor. This cofactor is called the NLP1 or neurophilin that is expressed on human cells. In fact, the S2 protein, along with the rest of the viral particle, is binding to NLP1 to further gain entry into our cells. Now, so what happened, okay, if you stop, okay, if you are taking out of this cutting site between S1 and S2. So they use a mutant virus to conduct this experiment. If the virus does not have the site where furin can cut over, that means the entire spike protein stay intact, okay, it's connected, alright, the mushroom head is not losing in the process, the mutant virus cannot enter the cells, okay, that is one of the experiments they did and what they find out. Okay. They also further verified the importance of this NLP1 receptor, okay, by using an antibody to block, okay, the NLP neurophilin receptor that is on the human cell. By doing so, it effectively blocks S2 protein from binding to NLP1 and therefore stopping the viral entry. So that is two pieces of new evidence or new findings showing how important NLP is for the virus to gain entry. Now they further down go into a deeper look at what is specific that is interacting with the NLP. So they took out a small sequence of amino acid and then they synthesize it and put it on a silver nanoparticle. Now this is a totally man-made setup, okay? A very tiny silver particle that is linked to a small peptide. Now this small peptide, I have the sequence here, I won't go into too much detail. The catch is that it has a C terminal. That means it has a carboxylic ending. Now having this ending is important for it to interact with the NLP1 receptor, and in fact through this experiment this small peptide facilitated the binding with the NLP1 and therefore draw the whole silver nanoparticle into a experimental human cells. Okay, so all three pieces of evidence here combined show you how it facilitates the entry of the virus. So what is the take-home message of today? Now the NLP1 neurophilin can be a cofactor for viral entry in cells with low levels of ACE2 receptors. So having ACE receptor alone may not be the whole picture for viral infection or viral entry into the cells. And the virus enter the cells can be stopped, okay, by blockade of NLP1. And they also find out specific amino acid sequence that facilitates cellular uptake of the entire virus. Okay, so therefore the end message is that the NLP1 could be a novel target for designing new antiviral therapies. Now to learn more, here is just one article, okay, that is published a couple days ago on Science, okay, in the Science journal article, neurophilin 1 facilitates SARS-CoV-2 cell entry and infectivity. Okay, now this is a in vitro experiment. It's done in the cell. I hope there will be more coming up in the near future showing a little bit more animal perhaps type of a study. Okay. So I hope you find this video informative. I'm really eager to see more research and more paper in this area talking about this new target that we can use to fight the virus. Okay, now that is all for this week's COVID-19 update, and I'll see you next Sunday at 7 p.m. again for another update on COVID. Bye.